Good morning. I was going to give the subtitle of this presentation would be Collections, Collaborations, and a Mistake I Made, but then I realized I could say Collections, Collaborations, and Confusion. So as Joanne alluded to, we do collaborate with lots of people in our research, and you'll hear a lot about that. The people I've worked with on this project is Becky Brown, who has the enormous job of trying to manage the invasive species in the province. Dr. Allen, where we did DNA work at the University of Victoria. Laurie McCormick, a fabulous technician who just cranks out data faster than we can analyze it. And Joanna Jansen, who used the ruler that you will learn a little bit more about. She's now a graduate student in Florida. So this project began with a request from the manager of a website called E-Flora at UVIC Geography Department. And the question was, does the Royal BC Museum have alien common reed? Alien common reed is a European plant. It's not native to North America, and it's been given its own subspecies. And as the story unfolds, you'll find out how difficult it is to distinguish it from the native North American version of the same plant. So here's a couple of our specimens. Here, the reason we have multiple specimens for each species in our collection is to answer this exact question. Where does such a species occur? And in this case, where does the alien subspecies occur? Our oldest specimen was collected in 1897 of this species. It came from Surrey. Don't hold that against Surrey. And I had actually encountered this plant in 2004 on a field trip with Dr. Allen at UVIC. We were collecting actually in Oregon and Washington. We were collecting alpine plants, but we were crossing the eastern Washington. We drove by this stand of this ginormous grass. This is a type of grass. And neither of us knew what it was, and so we did what all biologists do when they don't know what something is. We took a photo and we collected a specimen. So this plant is now in our collection. A specimen of this plant is in our collection. So what's the problem? Why is this? What is this concern about distinguishing the North American subspecies from the European subspecies? And just have a look at this. That kind of tells you the story. That is the invasive European subspecies. And it competes more strongly with native vegetation and wetlands than any other invasive plant. So if some of you might be familiar with Lythrum salicornia. Erica.
Snappy?
Lythrum salicornia, purple loosestrife. Purple loosestrife, and this plant will outcompete purple loosestrife, which is a significant weed in wetlands. And if you go on the web, you will find all kinds of comments. If you just ask Dr. Google, Phragmites or common reed, you will get lots and lots of comments. Now the two subspecies are distinct in some regards, but they're very difficult to tell apart, and that's part of this story here. And note this comment here, it can be difficult to distinguish between native and invasive subspecies. So the story for this project actually began in 1992 when someone collected a specimen from Metchosin. And it was identified by our former curator, Chris Brayshaw. He identified it to species. And subsequently, the Canadian expert for distinguishing the invasive and the native species, native subspecies, Paul Catling, came to our collection. Actually, more likely, we sent him the specimen in Ottawa, and he identified it. He confirmed, yep, that's the right species. And then in 2011, some guy named K.L. Marr, who had been asked by the web manager for E-Flora, said, okay, I think this is subspecies australis, which is the introduced European subspecies. So that prompted Becky Brown, the invasive species manager for the province, to go to the owner of Metchosin and say, we need to eradicate this plant from your property. And the owner of Metchosin said, no, I don't think it's a weed. I think it's been here a long, long time. I don't believe that it's been correctly identified. Whoever K.L. Marr is, got it wrong. And so a bit of a controversy developed, and on his website he made some comments. And so about this time I was at a meeting in Richmond with invasive species managers from all over the province, and I met Becky there, and I also met a young man who had sent me a specimen from Richmond that he suspected was an invasive type of common reed. He came from Quebec, and he said, we are spending thousands, tens of thousands of dollars controlling the spread of this plant. Do you think this is the native type or the weedy type? So Becky said to me, can you help us distinguish these? Because Paul Catling, his lab was no longer doing this work. So I said, yes, I knew by that time that this paper had been published that the native and the invasive genetic type subspecies could be distinguished using DNA markers. So this is a little box, very critical for DNA analysis. You put in a tiny amount of DNA and it amplifies the DNA and you get a huge amount of DNA out of that box. Now that requires a hot springs bacterium because it's an enzyme in that bacterium that is critical for this process, this amplification process, going from one strand of DNA to exponential number of DNA strands. So that's one of the critical things for DNA analysis. So all of molecular biology depends on the enzyme from this hot springs bacterium. And if you think you're a superior organism, you try seeing how your enzymes function at 90 degrees Celsius. So we knew from the literature what the DNA sequence of small regions of chloroplast DNA looked like. Here's the native type, the invasive type. You can get that information from the National Institutes of Health in the US. They have a huge data bank of DNA sequences. And note this difference here. So the Metchosin sample has an A. These are the 4 different abbreviations for different DNA bases. So this matched up with the native type, not the invasive type. So I was wrong. The landowner was right. So then this launched a province-wide study of this species. So Becky Brown, actually we were all together in the lab at UVIC and we were like, oh my gosh, how's it going to align, how's it going to align? And it lined up. The Metchosin sample was the native genetic type. The Metchosin sample that I had said was invasive. So here you see what's called a haplotype network. It describes the genetic relationships among different genetic types. Each gap separates one genetic type from another genetic type. And right there is the division between the native genetic types and the introduced genetic types. So now that we knew which genetic types occur in the province, then we could map them. So here you can see a map. Most of the province has the native genetic type, but there are some introduced populations as well. The really problematic one is right there because we analyzed 10 individuals per population. That's a single individual that's the invasive genetic type. It's in a population that's dominated by the native genetic type. There's the possibility of hybridization, and maybe in the discussion we can talk about what the implications are for that. So this is sort of an expensive technique. It's not real practical for a land manager that wants to know what they have. So we thought, let's see if we can use just some basic observations of the flowers, the seeds, the leaves, the stems, the inflorescence. Are there any other features we could use to distinguish these 2 genetic types from each other? So this is where the ruler comes in. Here's a flower. That is a grass flower. It's in full bloom. Flowers don't have to be pretty. The function of flowers is to produce a seed. And all of you today will have consumed or will consume the products of grass flowers, except you called it oats or barley. You'll have maybe a fruit drink tonight called extracted from barley. Corn, all of these are rice. These are all grasses. And they all come from flowers just like that. So you can do measurements on all kinds of features on the plant. So what we did, we thought, okay, there's a number of measurements that have been attempted in the past that didn't distinguish the 2 types. Are there any other measurements we could do? Turned out not really, and I'll show you the next slide. So even this little flap of tissue here, the native genetic type, if the length is greater than 1 millimeter, it's the native genetic type. If it's shorter than 1 millimeter, it's the introduced genetic type. Very difficult. And here's a basic specimen. We could measure the leaves. We measured several leaves. We measured everything we could imagine that we might be able to measure that might be useful for a landowner and land management people. So this is what we got. Measurements from 48 specimens. And all you need to know is each one of those boxes is a different feature. Doesn't matter what it is. Different part of the plant. What you do need to know is see the overlap between native and introduced. Just about every feature you could measure, there's a range of variation. Yes, out at the ends of the bell curve, there are some distinctions like this color class, and there's a difference between native and introduced type there. But for the most part, this is a significant amount of overlap. So this is the final summary, is just to say, in many cases, the identification of whether it's invasive or native is going to rely on DNA markers. Thank you.